So yes, my name is Josh Arens. I'm a part of the Weymouth group at Stanford. And today I'll be talking to you about charge-altering releasable transporters, which are a polymer that can be used to encapsulate genetic material and then delivered into cells. So as I'm sure many of you are familiar, last year the Nobel Prize in Physiology or Medicine was awarded to Catalin Carico and Drew Weissman for their discoveries concerning nucleoside base modifications that enable the development of mRNA vaccines against COVID-19, which Lee Jun discussed like their great impact on the world in the last few years. And this discovery paved the way for those mRNA vaccines, but also new types of gene therapies like using CRISPR-Cas9 to get into cells and to like actually edit the DNA itself, or gene silencing where siRNA interferes with the expression of certain mRNAs inside the cell. And there's some, like with the CRISPR-Cas9, there's some drugs in clinical trial. And then regarding siRNA, there's available, like commercially available drugs like Onpattro for different types of diseases. And so lipid and polymer nanoparticles are two different types to deliver genes into cells. So we heard at length kind of about the LNP world and the different kind of molecules used to make up LNPs and encapsulate DNA or RNA and get it into cells. Today I'll be focusing on polymer nanoparticles. And so there's a couple, or well, several different types. Two bigger types are both amino esters, and above we have polybeta amino esters, and it's just beta because there's two carbons in between this ester and the nitrogen here. And then below we have an alpha amino ester. There's just one carbon in between the nitrogen and the ester there. And that's what I'll be talking to you all today about. So CARTs, it stands for charge-altering releasable transporters. And CARTs are an amophilic co-oligomer. So we have one block. This R1 is going to just stand for lipid. And then that block will be lipophilic or hydrophobic. And then this other block is going to be charged in aqueous conditions and is going to be the part that interacts electrostatically with mRNA to form kind of this messy nanoparticle. The other unique thing about CARTs, as is kind of evident in the name, is that the charge alters under physiological conditions, and it's through a chemical process. And so, as I mentioned before, these are blocks. And so the nitrogen here, it's going to be next to another like monomeric unit just like it, and it'll kind of like bite itself and go through this mechanism where it'll eventually neutralize to form this diketylpiperazine structure, and then it's leaving behind this kind of intact lipid block. And then now all of a sudden the thing that's electrostatically binding the mRNA, it's like neutralized, and that the theory is anyway that helps release it into cells once it's inside. We've also discovered over the years that the chemical structure of the polymer itself really affects things. So on one end, we can change the lipids. We can use different types of lipids, and that'll affect the efficacy, like how much RNA gets into the cell, how much is released. It will also affect the selectivity. So we can tune which types of cells we get into based on changing the lipid blocks. I won't discuss that too much today, but there's some early work on that in our group. More recently, we've kind of been looking into how changing the cationic structure affects organ tropism. And so if R2 here is just like a proton, then we call that like a glycine-derived CART. I'm not going to go into the reaction scheme up there, but these are all derived from amino acids. And if it's like glycine-derived, we've found that like it almost exclusively goes to the spleen in mice, like when you deliver by an intravenous injection. If it's lysine or ornithine derived, which means there's going to be like it's dicationic per monomeric unit, right? We have 2 protonated amines per unit. That almost exclusively goes to the lung, as you can see here. And then if it's like tyrosine, it has this kind of polar functional group attached, then it goes to the liver. And so it's kind of this unique platform where we can change, like we can see very directly how chemical structure affects biological function.
Yeah, why is that?
Oh, short answer, we don't know. There's theories about that the chemical structure is going to change which proteins like form a corona around it. And so we've been kind of looking into that, and we've actually done some SAXS where we, essentially dip our nanoparticles in proteins and see if that changes the structure, and it doesn't seem to do too much.
Are you gonna show us that?
Not today, actually. Yeah.
It's like 20 amino acid or just try 4 and then make some explanation on it?
So our approach has been kind of like we've just tried different amino acids in like different types of amino acids, right? So we've tried like more hydrophobic ones or basic ones or whatever. And then so far we haven't done enough to like see specific trends based on that. We have a postdoc who kind of hypothesizes that there might be some stuff, like for example, the 2 dicationic amino acids, they both go to the lung. And so there might be like, but I assume that just has to do with the charge density, because that's pretty well known in the literature. Nanoparticles that are more like positively charged on the surface tend to go to the lung. But yeah. And so today I'm kind of going to focus on like what these CART mRNA nanoparticles actually look like, since we've used cryo-EM and SAXS in tandem to kind of get a good picture. Because when I joined the lab, we really had no idea what they actually looked like. But in the last 3 years, we've made like a lot of progress and I have a pretty good idea of what they look like, and then also how that structure affects their function. And so for these examples, our early examples, we used an ornithine-derived CART, which is that dicationic one that goes to the lung, and then we would just formulate it with mRNA. And here we did it at NP ratio 5:1, meaning there's 5 times as much positive charge as there is negative charge from the mRNA. And then here we did it 10:1 NP ratio. And you can see we kind of have these kind of blobs. They're roundish, but they're not really perfectly spherical, not like the LNPs, which have these really well-defined shapes. And in terms of like internal structure, right now we're kind of calling it like this bicontinuous structure. And then we kind of see like on both SANS, small angle neutron scattering, and SAXS, that we get this feature that corresponds to about 6 nanometers. And if you were to take like snapshots of the cryo-EM images and like measure the difference between the light and dark regions, it corresponds to about 6 nanometers as well. So that was a nice early result.
The dark, sorry, I think I had.
No, all good.
You go ahead.
Okay.
You will probably respond to that question.
Were you asking if like dark is hydrophobic or?
I was thinking about the RNA. That's the darkest RNA. Do you have indication for it? Or it's just the hydrophobic part?
Our thought was that it was the hydrophobic part, but that's just kind of a guess. I don't think we have any firm evidence one way or the other. And so here's just like a comparison of like this early kind of CART mRNA nanoparticle to different LNPs. And again, as I mentioned before, like with the LNPs, you kind of get more highly ordered structures generally. But with the CARTs, at least the CARTs that we've tried with mRNA, we really don't see them. Again, it seems to kind of be this bicontinuous structure that generally forms like a round shape. And then, as I mentioned before, like chemical structure affects function. We also see that it affects the like structure of the nanoparticle itself. And so if we look at kind of yellow and green These are both glycine-derived carts, meaning the cation is the same. All that we're changing is the lipid. So in the yellow, that's a dodecyl lipid, which is a saturated lipid. There's no double bond in it. And you can see that like the structure on SAXS is a lot smaller, like it corresponds to 6 nanometers. We kind of expect that the reason for that is like the saturated lipid allows it to pack tighter. Whereas here you have 2 different unsaturated lipids with a kink in the chain. And on top of that, they're different sizes, right? This one's 18 carbons, that one's 9. And you get larger, like a broader feature. And on top of that, a feature that corresponds to 9 nanometers, suggesting like the spacing is larger. And then below, we also tested like a lysine-derived cart and an ornithine-derived cart, both of which are dicationic, both of which also have that saturated lipid. And they're even a little bit tighter packing than the dodecyl glycine cart in yellow, right? They're both a little shifted to the right. And then here's just some cryo images showing like these examples here of literally looking at the difference between the kind of light and dark regions. And you can see on average, it does seem to correspond pretty well to what we're seeing in SAXS. And then one of the projects I've been working on is looking at how polymer sequence might affect the structure of the nanoparticles. Because up to this point, all of the carts I showed you were block carts. So all the lipid units were on one side, and then all the cationic units were on the other side. And that is kind of shown here in this doodle. And then what that results in, right, once it's in an aqueous solution that has a pH about 7, it's like all of your cation is going to dissolve into that, or sorry, not dissolve, but degrade into diketopiperazine, leaving behind the intact lipid block. But we thought, what would happen if we just polymerize these statistically? And I'm not going to go into the chemistry, but we did some kinetics showing that when we do a statistical copolymerization, meaning we polymerize both monomers at the same time, the lipid monomer is a little faster. So it ends up being gradient instead of blocked. And so it's not a huge, like it's not like a random copolymer. It is a gradient copolymer, but it's, you're still going to like get some incorporation of your cationic unit in the lipid part. And so you might not get as clean of a degradation process. And so we kind of hypothesize that it might actually take longer to degrade because you're not going to always be guaranteed to have 2 cationic units side by side. That's like the only mechanism for that degradation. They could hydrolyze, but hydrolysis is a lot slower than the time scale of the chemical rearrangement. And so early on, we just did some dynamic light scattering experiments to kind of see if this affected the size or surface charge of the nanoparticles. And so here we just looked at dodecyl glycine carts, shown here. We made a short block cart, which is in pink. The total monomeric units or degrees of polymerization was about 25 for the short block, and also 25 for the short statistical cart. And then for the long statistical cart, it was 250, so an order of magnitude longer than the short carts. And what you can see here is like over time, like initially the short block cart has a average diameter of about 150 nanometers, right? It's in pink here. And then within an hour, it balloons up to like 400. And we associate that to the degradation of the cart, right? As it degrades, as the charge on the surface becomes neutralized, it can aggregate with other particles because they're now neutral particles. And so we're kind of taking the change in size as an approximation of like the degradation of the cart itself. Whereas the statistical carts, they're pretty constant up to like 2 hours. And so that was a really interesting result. We kind of see that replicated with surface charge, although it's a bit of a like you see some change in surface charge right away, but it's a lot slower than it is for the block CART, right? Because they start off as positively charged on the surface, and then over time, as they degrade, they neutralize and eventually become negative. And then another interesting thing is that the polymer sequence affects in vitro efficacy. So again, when I kind of joined the lab, we were told that you couldn't make these CARTs longer than about 25 units long, that if they were longer, they didn't work. And you can see that here in blue, right? The long block CART does not really get RNA into the cell, or maybe it does get it in the cell, but it doesn't release or something, like it's not expressing. However, with the statistical CART, you do see expression, like it's not as good necessarily as the shorter ones, but you still see that expression. And that's something we kind of wanted to explore through SAXS. And so in this first experiment here, we kind of compared 4 different CARTs. On top, in red and blue, we have 2 block CARTs, 1 short and 1 long. And then in yellow and green, it's 2 statistical CARTs, a short and a long one again. And so the block, we kind of see, as we saw before in previous experiments, this feature that corresponds to about 6 nanometers. And then both the short and long statistical CARTs, you kind of see that feature shift to the right, corresponding to like tighter packing of the RNA. Whereas the long block, you get this feature, and around there, it's going to correspond to like about 15 nanometers. And we kind of hypothesized, based on some cryo-EM images, which I unfortunately don't have in the slides, that's just like the polymer itself forming micelles, and that those micelles don't actually have RNA in them. And so that might be the reason like why the long block CARTs don't work. Yeah.
So when you're making these nanoparticles, what pH are you making?
Sorry. Oh, we're doing them at 5.5.
And only when you shift to 7 do they start this to come apart?
Yes. So I've done some like DLS showing if you keep the pH below 6, that they'll be stable for like several hours. That the size and zeta potential don't really change. There's some degradation happening because, as I mentioned before, they can hydrolyze, but hydrolysis is going to occur much more slowly, like orders of magnitude more slowly than the chemical rearrangement. And then we also see that the polymer sequence affects nanoparticles in different types of CARTs. So here's a mixed lipid CART. Before it was the saturated CART, here it's 2 unsaturated lipids. Interestingly though, so this we don't really know how to explain or like why it's the case, but when you compare the short block to the short statistical, in this case, like again, you see a shift to the right, suggesting like packing's tighter. But when we go to like long statistical, all of a sudden it looks very similar to the short block. And we can't really explain that. Interestingly, like when we did some kind of, we're working with a group at Stanford in the med school, and we're kind of working on this breast cancer drug. They found that the short block and the short stat part works better, the short block and the long stat work better than the short stat in their model. And so there may be something about the spacing in allowing it to kind of release into the cell or, you know, disrupt the lysosome. I'm not exactly sure, but it was an interesting result that we don't know how to explain at the moment. And then I'm not sure.
Maybe it's worth a second. Can you go back to your slides? Though, I mean, multiple sex curves.
Yeah.
Did you? So you comment on the despacing. That's proper. That's how it's supposed to be done. Do you ever try to figure it out the secondary peak at the higher Q range?
Up here.
Because even that you have a different despacing, they're showing up in the same position.
Yeah, we kind of guessed that that's probably like the RNA itself, some feature in the RNA, but I'm not totally sure. We haven't looked into it too much, but you're right, like that feature stays the same regardless of what these are over here. And so we thought it might just correspond to like the spacing between the RNA or like, you know, but yeah. And then, okay. So we also began looking at like what would happen if we changed the cargo itself. And so all the examples I gave before were related to our mRNA, but one of the projects we're working on, we are using siRNA to kind of knock down an oncogene. And so we wanted to see if that has an effect on the structure. And like when you actually compare mRNA to siRNA, you would think it would, because mRNA, right, is like 2,000 nucleotides long and it's single-stranded, whereas siRNA is only 20 nucleotides long and it's double-stranded. So you really have kind of like this, the siRNA is going to be more like, you know, bricks or rocks, like they're going to be chunky units, whereas the mRNA is going to be polymeric, right? It's something that's going to be flexible. And interestingly, we did see like a pretty big difference between the two. And so here are, in kind of green and pink, are just two different carts we used. Above is the glycine-derived mixed lipid cart, and below is the ornithine dodecile cart. And then in kind of the darker color is with the siRNA, whereas in the lighter color, it's mRNA. And with mRNA, we just have the one peak, whereas with siRNA, it's a little messy here, but you do see two peaks, two features, one that's higher than the other. And then you don't exactly see the two features here, but if you squint, you can kind of see like a broader thing with a peak, a sharp peak in there. I'll hopefully convince you by showing you the cryo-EM images as well. And so again, in the lighter color, that's the kind of ornithine cart with the mRNA. And then on the left here, we have the ornithine cart with the siRNA. And I'll make that bigger. We actually kind of see two phases here. And so if you look on the edge, you kind of get this lamellar thing on the surface. And then in the middle, you kind of have that bicontinuous phase. And so that's kind of what we're associating with that kind of sharp peak at like 5.9 nanometers, and then the broader peak at 6.2. And we see that with other types of carts that we tested with the siRNA versus mRNA, these kind of like this, these two features in the SAXS data. And so just to kind of wrap up, we saw that like the sequence does matter when it comes to the properties of the nanoparticle, which include degradation that we measured by DLS and ELS, and also kind of the spacing between the hydrophilic and hydrophobic layers in cryo-EM and SAXS. We also saw that it has an effect on the like biological function, where polymers greater than 30 units don't work if it's blocked, but they will work if it's statistical. We also saw that the genetic cargo matters, right? mRNA, we just kind of see that one feature that we call it the bicontinuous packing, whereas the siRNA, we get more complex nanoparticle with lamellar packing on the surface and then kind of bicontinuous packing in the center. And then future directions, we'd like to see how like NP ratio might affect structures, because a collaborator of ours recently showed a type of cart that when you change NP ratio, like it's the same chemical structure, if you just change the NP ratio, it'll go from lung to spleen. And so we kind of want to look into that. We're also interested in time-resolved SAXS and or cryo-EM to kind of see how the structure changes as it degrades. Yeah, we'd just like to thank everyone in my group, the SIBYLS staff, Greg, Aimee, and Josh, and funding sources from NSF, NIH, and the Center for Molecular Analysis and Design at Stanford, and happy to take any questions.
Yeah, you guys have a blue coat.
They're flame, like fire resistant.
Fire resistant.
Yeah.
Other questions?
Yeah.
Yeah, so those cryo-EM look
So, yeah, there's a postdoc in the lab who does the cryo, Paul. I think you met Paul actually once, maybe. But I think he has done the FTT of the cryos. And I think it does look like, I think it does correlate well to like what we're seeing with SAXS, but I'd have to
Yeah,
check
I
with
mean,
him.
just a quantitative way to make sure the feature and stats is matching with what you think.
Yeah, for sure.
Quick question. So in terms of your analysis, are you guys feeling that your analysis is quick? Is there anything that needs to be developed for your analysis of the data that you're getting from us?
Yeah, I think the analysis works well because we just use that like website to average all of it, and then we have this like Python code that we use to just make it into graphs easy now. But yeah, I think if anything, we're interested in like trying to figure out a way of getting it. So like, and I think part of like since we come up already to formulate, we're trying to figure out a way of like maybe getting it somewhat time resolved, so we could see or like can change the conditions of the formulation to like see it degrading, and if that's going to affect like the SAXS that we get. I'm not sure, but we could talk about that more at some point. But that's something we are interested in doing.
Yeah, I mean, we could set it up so that you're basically running in and out of the hutch.
Yeah.
On time, because your time resolved is more like minutes, not seconds or
Yeah, although we
milliseconds.
can kind of tweak it because like I played around with the pH and there is some wiggle room where you can make it longer if you need. But yeah.
But it's not like milliseconds. Like
No,
when
yeah,
people
it's-
talk about time resolved, sometimes they're like,
No, yeah, this is like minutes to hours, I'd say. But
okay, well, we certainly can make room for that kind of experiment. Like you guys come for a day and are just like
yeah, okay.
running. It'll be an athletic event.
Yeah.
I have also a very short question. Are you interested to separate different sizes? I mean, this is pretty heterogenic from the curve that we see. I mean, that's
Yeah.
why you haven't shown that in cryo-EM. But do you think there is some populations that you're interested, like super small or super large?
Yeah, so that's something we're kind of looking into is like we're starting to get the impression that like the NP ratio we do affects the size of the nanoparticle. And so like we've seen that like lower NP we get larger nanoparticles, larger NP we get smaller nanoparticles. And we tried to figure out ways we could we assume when we have a high NP that we're going to have some kind of like polymeric micelles that don't have mRNA, right? Because like it's 10-to-1, like by definition, there'll be some that's not a part of the nanoparticle.
Do you want to separate that out of?
But right now we don't know how at all. Like
We had potentially some solution connected with the SAXS. We just ordered a F separation,
The field flow fractionation.
split flow fractionation.
okay. Yeah, no, because that's something we've been brainstorming about is seeing if we can try to filter out, the other particles, because there's clearly other things in there.
And when you were doing the comparison of the mRNA and the siRNA, why was the SAXS curves for the siRNA so lazy? Or something was different about that?
Yeah, I think that day the needle was having issues, like, because it wasn't, and it was like forming bubbles and things, because I remember they had to run the plate like a couple times because there was
So
some issue.
It's not something inherently different about siRNA.
I don't think so. We're hoping to run it again, just to confirm. So hopefully in the next cycle. But yeah, I think it was just that day because previously we had shown, like, we did one sample in a run before that and it looked fine. And so I think it was just a batch issue.
Yeah, probably just, they were able to select one frame out of the.
Right, that's kind of what it looks
Yeah.
like
Yeah, rather than all of it.
just less data.
All right, thank you again, Josh, and